Assalamu alaikum dear students. Today our topic of discussion is tertiary structure of proteins. The tertiary structure refers to the specific three-dimensional structure of the polypeptide chains of the proteins. Detailed knowledge of the three-dimensional structure of proteins has been made available by the application of X-ray diffraction methods. Once a polypeptide chain has achieved secondary structure, it continues to fold and finally is packed into a compact, almost globular three-dimensional shape, which is called its tertiary structure. As a result of the formation of secondary structure, the side chains of each amino acid in the polypeptide is positioned in a specific way. If additional hydrogen bonds, covalent bonds, hydrophobic interactions, or other atomic interactions are able to form, the polypeptide folds. This polypeptide folds to accommodate them to attain its three-dimensional shape. My dear students, this polypeptide chain, when it undergoes coiling, folding, it leads to the formation of tertiary structure. The way the structure folds has an important bearing on the properties of proteins. The folding brings together active amino acids which are otherwise scattered along the chain and may form a distinctive cavity or cleft in which the substrate is bound. In protein consisting of a single polypeptide chain, for example myoglobin, the tertiary structure determines the overall shape of the molecule. Thus, proteins are called globular proteins if they are very compact, and fibrous proteins when they form long, thin threads. The tertiary structure is determined by a number of bonds which are of different types. For example, disulfide bonds, hydrogen bonds, ionic or electrostatic bonds, hydrophobic bonds. What are these disulfide bonds? These are bonds, these are covalent links which give some rigidity to the protein molecule. They are relatively more stable than other types of bonds. A disulfide bond is formed between two cysteine residues. Likewise, we have hydrogen bonds here which are very weak, but since they are numerous, they give considerable stability to the protein molecule. Hydrogen bonds can be formed by sharing hydrogen between amine, nitrogen and carbonyl oxygen of polypeptide backbone. Likewise, we have ionic bond or electrostatic bond which are formed when an acid or ionic amino acids are ionized and lie close together. So we have hydrophobic bonds which are formed because of tendency of non-polar side chains of neutral amino acids to closely associate with one another. When protein molecule is submerged in an aqueous medium, there is tendency to expose a maximum number of its polar groups to the surrounding medium. Conversely, a maximum number of non-polar groups are directed internally. Although the hydrophobic interactions do not form a true bond, they are of great importance in the formation of tertiary structure. So it was all about tertiary structure of protein. You can have a look here, one more